Hi, and welcome to the very first video in our series on pollinator identification training for insect surveys in Boulder and Longmont for the Neighborhood Nature Lab the summer of 2026. My name is Andy, I am with Butterfly Pavilion, and I'll be going over some of the introduction and methods in this video. Please watch the rest of the videos in this series to tackle each of the taxa and learn how to identify them. First, let's talk about why we need you guys to help us study pollinators. As you may or may not be aware, over 87% of all of our flowering plant species, many of those important food crops, rely on pollinators for that seed production. Without our insect friends, they don't get pollinated and they don't become fruits, which we eat. And conservation biologists estimate that general invertebrate populations have declined by as much as 45% globally over the past 4 decades. And that figure does come from the Druzo et al. 2014 paper that I have cited there on the right. If you'd like to check that out, it's a really cool paper. Even just in Colorado, we are worried about the Suckley's bumblebee, which is a cuckoo bumblebee that relies on the western bumblebee as its host. We're worried that it may already be extirpated from Colorado, and what that means is that Colorado was a part of its native range, and it is no longer found in the state of Colorado. So it's not extinct. It may be found somewhere else, but we know it's not here anymore. This is part of why we need volunteers like you folks to help us with this long-term monitoring. Research scientists love this work, but we cannot be everywhere at once. And one of the excellent parts about community science is that we don't have to be. We can recruit the people who know the area best. You guys are out there on your walks in your neighborhood. Maybe you're walking the dog, maybe you're taking the kids to school, and you see your neighborhood every day. And that's something that we just can't do with the number of scientists we have out there. So that's part of why we need your help. A part of the study will be comparing more developed locations in the cities directly in Boulder and Longmont to some more intact plant communities that we're finding out in areas like Button Rock Preserve, where they've been a little bit less disturbed. There's a little bit less urban development in those areas. That helps us to establish some of these baseline levels of insect biodiversity and figure out sort of like an inventory. Who lives here? When are they here? What does that inventory look like? And that baseline is really important to not only examine the effects of urban development, but also to understand prior to any restoration work, we have to know what we've improved from. So developing these community science efforts local to you as well, we really hope will help us achieve that and also help you all understand a little bit better what's in your backyard. We have current locations for survey sites in the city of Boulder, in the city of Longmont, and at Button Rock Preserve, as I mentioned, which is out west of Lyons. That is an area for survey that we will be taking out volunteers with us, with staff members, just because that is gated off, and we don't want to ask you to hike several kilometers with all the gear that we're going to ask you to bring. As a part of this survey, we're going to ask you guys to use iNaturalist. We're hoping that this is a relatively accessible way for you all to take pictures right in your own backyard. If you haven't made an account already, I strongly recommend it, even if you don't end up following through with this survey and volunteering with us, because it will help identify things right on your phone. Like the image on the left, it will sometimes offer a suggestion identifying what's in that image using AI, and that AI does get better and better the more that we train it to do so as we take these pictures and identify insects. So it's a really neat app just to have. But it's also going to be really easy to submit that data to us if you enjoy that app. So if you join the project, for which there will be a link underneath the video description, called the Neighborhood Nature Lab, if you join that project and you are within the areas that we are studying, you should not have to do any extra work to upload that data to our project so that we have access to that study data. If you are in the right location and you make an observation that is not a vertebrate, so if you have a deer in your backyard, that's really cool, but that won't upload to our project. If you have a picture of bees, however, it should automatically get added to our project if you've joined the project and if you have that picture in the right area. So it shouldn't be any extra work on your part, and it is a free app. I do know that there is both the classic version of the app and a newer version of the app on whether you have iPhone or Android, and I personally have the new version of iNaturalist, and I know some people prefer the classic version, both versions, there should not be any issue no matter what version you have. Both versions should allow you to submit data and be relatively easy to use the same way. So there shouldn't be a difference between the two of those. So where are we asking for volunteers to help us survey? We have a couple of sites designated in the city of Longmont as well as in Boulder, and those will be on 2 different links depending on which city you're at or would like to volunteer in. And those maps will outline the general area and a recommended parking spot and recommended walking path. Now, different things are going to be blooming at different times of the summer. So if you see something along a related trail that's in the same general area that's highlighted, please go ahead and take that trail. You don't have to take the recommended walking route. There are some trails even that Google would not let me recommend as a walking trail because it didn't recognize it as a walking trail. So please take this as a general guideline and just stay within the overall area where we want to make sure that we're in areas that we have permits to be in. If you have any question about permits, please reach out to me at adraper@butterflies.org. Let me know where you'd like to volunteer, and I will make sure to get you a copy of that permit to carry with you while you're out there. If you're, going off the trail a little bit and taking pictures of bugs to get to that flower. So go ahead and email me if you have any questions about those permits or any of those maps. Those are all using Google Maps, so they should be able to be pulled up on your phone with you in the field to take a live look at where you are. So you've got your map, you've got your iNaturalist app. How are we going to go about collecting data? There's going to be 2 different ways that we're going to ask you to collect data. That first system is going to be a tally system, just on paper, how many times is this type of insect visiting this type of flower. And another method that we're going to talk about using photos is going to follow a methodology that will be very similar to you if you have looked into the Xerces Bumblebee Atlas or Bumblebee Watch, where we are going to cool down these insects and take much more detailed photos to upload to iNaturalist. For that first methodology, those visual surveys with tally marks, we're going to ask that you guys are staying within that general area on the map, on a trail, and just sort of meandering freely from different flower to different flower. If you're not seeing anything exciting visiting those flowers, that's also okay. That's still important data. We still want to know that. So please do record the different types of flowers that you're seeing. We're not asking that you be 100% in your plant identification. If what you would like to do is take a picture of each of the different types of flowers that you're seeing and upload those to iNaturalist, that's a great way to get a quick identification on what types of flowers that you're seeing. So it's okay if it's a general, sunflower, no visitors, no tally marks, that's fine. General dandelion, that's totally fine as well. If you're not seeing anything visiting those flowers, feel free to keep walking on to the next group of flowers. And include those observations to the level that you're confident in identifying. You guys will be doing a lot of training here in the next several videos as we learn about each of the major pollinating taxa. But make sure that even with all your resources, insects are quick. If you can only say, okay, I think that was either a bee or a wasp, that's much more helpful than just taking a guess. So record to the level that you're most confident. If you see something and you're like, I don't know what that was, despite all the training that I had, but I know it was a beetle. That's perfect. Just write down beetle. That's much more helpful than a guess for something more specific. And if you'd also like to stop and refer to any of the identification resources that we will link to, please do so. Like we're encouraging you to use all of the resources at your disposal. This is this is not a closed book exam. Feel free to go ahead and do that, and then subtract that time from your total survey time, because we will ask you on that worksheet what time you started and what time you ended. Just know, minus 5 minutes or whatever, because I brought out my guide and I stopped for a while. So we won't count that towards your total survey time, because we want to encourage you guys to use those resources. In this next slide, we have a copy of the worksheet that we're going to ask you to fill out as a part of those visual surveys. At the bottom in red, you can see the types of flowers listed on this hypothetical walk, and on the left in that first column, you can see some of the major groups of bees that you might see. So making this a super easy tally system, if I see one hairy leg bee on a sunflower, I can do 1 tally mark. If I see a bunch of tiny dark flies on a white flower, I may not be sure what that white flower is yet. I can write that down And remember to come back and take a picture of that, upload it to iNaturalist, get a quick identification, or use my field guide, or any number of resources as well. So if you do something like that, please feel free to go back, use your resources, and write in the best name possible. Or if you do take a picture and upload it to iNaturalist, you can always write in the notes of that picture, this goes with, this survey on this date. That kind of information we can always link back together as well. At the top of this worksheet, we'll see all of the site information. So for this example, I said I was walking in the Heatherwood Park in Boulder. Maybe I'm surveying from 3 o'clock to 3:30, and maybe it's partly cloudy with a little bit of wind. So I've got my name, the number of volunteers, all of that site information. If you don't mind including that at the top, that's going to help us fill out a lot of our survey data as well. So on that tally sheet, when am I going to record flower visitors? What I'm going to record is when I see that insect either touching the petals or the inside parts of a flower, as seen on the image on the left and the middle here. Those would both earn a tally mark on my worksheet. On the right here, we can see a fly on a leaf, and in that case, that would not count as a flower visitation for my observation. I also mentioned that we're going to be taking pictures of insects as a part of this survey. So let's talk a little bit about insect photography. First, we'll do a couple general tips, and then we'll talk about our survey methodology. So when we're photographing insects, whether it's for a survey or just for fun, one of the things that really helps that photograph come out really nicely is a simple background. For purposes of a survey, we can recommend like a piece of paper, like the data sheet that you're carrying, maybe a field guide that you're holding. Something like that in the background can really make the photograph much nicer to see and can help sometimes the camera focus on the insect itself. If you're doing this just for fun and you're editing it later, a simple background as well can help make that photograph come out really nicely. One of the other things is don't be afraid to get too close to these insects. Some of them may fly away, and that's fine. But I have come up to bumblebees before that have been so invested in these flowers that I have put nets over them. I've put the camera directly in their faces, and they just did not care in the slightest. They had a job to do, and they were going to do it. So don't be afraid to get close to some of these insects. They're not all going to fly away, and you'll get some good photos out of it. For purposes of our survey, we're going to take some of these bugs and cool them down. And once they're nice and chilly, they're not going to move. They're for lack of a better phrase, cold-blooded, right? So when the environment's not warm, they're not moving that much. And they have no interest in stinging you. They have every interest in warming up. They are not going to be interested in harming you at all. So don't be afraid to get that camera right up in there and to take that photo. If you are using a camera with a macro setting, use that setting, utilize that. That's going to help get those really up-close photos in focus and highlight those features. If you're using your phone camera, some of the new Androids and iPhones do have a macro setting, as well as there are available some macro lenses, either online on Amazon or whatever. And we have a couple of clip-on macro lenses that schedule ahead of time with us to swing by, and we have I think 4 currently available just to clip on to your camera on your phone to help you with that macro lens. Once you're done using those for your survey, go ahead and return them so that other people can borrow them, of course. But also make sure that there is lots of light. As we focus on a smaller and smaller point with our cameras, there is less light reflecting off of that object to bounce back into the camera lens, right? So a high amount of light is going to really help that image come through. Okay, for our photo surveys, we're going to walk through the designated survey area just like we were for our visual surveys, and we're actually going to use our butterfly net, and we're going to collect all those flower visitors that we see. Take those animals and put them in a jar or a vial. This can be something you have at home, whether it's like a jelly jar that you've washed and rinsed out, or if you have a prescription pill bottle that you can take the label off and you can see through it. Those make really nice vials. Anything like that at home, or if you would like to borrow some from us at Butterfly Pavilion, some glass vials, go ahead and shoot us an email, and we can schedule a time and get some of those resources to you. But carefully put that animal into a jar or vial. and place that vial in the cooler relatively quickly. Once we have a bug in a jar, we've effectively trapped it in a greenhouse, right? So that light's going to come in and heat up the inside very, very quickly, much more quickly than we might realize. So they're actually more at risk of overheating in that vial than they ever are freezing in our coolers, right? Our coolers are not going to get that cold that quickly. So let's stick those vials into our coolers with ice very quickly to keep that insect nice and cool. And when we're doing that, we can pause our timer, or if we have a timer going, or just keep track of taking a little bit of time off at the end. We don't want to count that against your survey time either. So go ahead and put those vials on ice for photographing for about 15 minutes. Getting your insect from your butterfly net into a jar can be a little bit of a skill, a little bit of a learning curve. If you're curious on how to do that, I have linked here a video on YouTube of Idaho Fish and Game giving us a great instruction on how to get that bee or insect from our net into a vial to get them into the cooler. So once we've had our insects and vials on ice for about 15 minutes, that animal should be chilled, not moving anymore. They may curl up their legs a little bit, looking like a deceased insect, but they're just chilly. We get some cold nights here in Colorado, and they will last through those just fine. So go ahead and place that insect on a flat surface. Again, we recommend something like the data sheet or a field guide underneath to provide that nice flat surface and a simple background behind it. And take a picture really closely of each of the different parts of the insect. So the head, the thorax, the wings, whatever you can get really cleanly on that camera. So each insect taxa is going to have different parts of the body that are most important for identifying it to species. For butterflies, of course, the wing patterns may help us identify it. For bumblebees, we actually also want to look at things like their legs. And for flies, some of the long hairs you see on flies, the tough hairs, rather than like short fuzzy hairs, can be important for identifying them as well. So there's a list of each of those characteristics by taxa, by group of insects, available in the written survey instructions that are in the provided resources as well. So if you would like a written list, that's available in a link in the description. It's always better to take more photos than you think you need. We can take a bunch of photos and just take some of the ones that are the best or most in focus and submit those to iNaturalist to help us identify the insect as best we can. When you're done taking photos, just allow that insect to warm up to the ambient temperatures, and when they're warm enough, they will fly away. So we've talked about how to photograph insects and how to record observations. What kind of time commitment are we looking at for each of these survey methodologies? What we would love to see, ideally, from each volunteer is half an hour of visual tally marks and half an hour of these photograph surveys. That totals for 1 hour at a site, and we're asking for once a month. If you're able to do that at the same site once a month across the summer, that gives us a fantastic set of data. If you're going out there with a buddy, we can also split that time with them. So if you're out there with a friend or a spouse or a kiddo or whoever's doing surveys with you, and you each do 15 minutes concurrently of a visual survey and 15 minutes of a photograph survey, bam, that's a whole hour when combined. So there are many ways to split this data up, but what we would love to see is at each site, hopefully there'll be a volunteer, and we can have at least 1 hour of data from each site per month. So in the next couple of videos in this series, we're going to talk about these 6 groups of insects as a whole. And in the next couple of slides, we're going to talk a little bit about some of these species that we would like you to be aware of as you are on your surveys. So while you're out conducting studies, we're going to talk briefly about some of the species that we know are of conservation need. And these are going to be classified by the Colorado State Wildlife Action Plan, or SWAP, that's put together by Colorado Parks and Wildlife and others. And these puts these animals into 3 tiers. So SGCN, or Species of Greatest Conservation Need. That tier 1 is also going to include a lot of animals that we'll see on the endangered species list as well at the federal level. We'll see tier 2, which may be, locally endangered but not federally. And then we have our species of greatest information need. So as we see invertebrates recognized as wildlife here in the state of Colorado, we're realizing just how much information we are lacking in terms of key data points, like the ones that you guys will provide as volunteers, to help inform decision making regarding the conservation of these insects. So this is just one of the many reasons why we need your help as volunteers to help collect more data about where these insects are at, what they're eating, all that kind of stuff. So let's cover a couple of tier 1 species that we'd like you to be aware of while you're out on your surveys. First, and probably most recognizably, we have the monarch butterfly. We know that the monarch butterfly has its important hosts on milkweed, and we do have some similar species in the area, including the viceroy and queen butterflies that do look somewhat similar, and we'll talk more about how to tell these insects apart from each other as we cover butterflies. But for now, be aware that we have the monarch butterfly in our area and that there are similar looking species in our area. These guys are active mostly from June through October and can be found throughout the state. We have several bumblebees in our area along the Front Range and throughout Colorado that are of particular conservation concern. This is the American bumblebee. I'm not asking volunteers to be able to recognize every single bumblebee species out there. There's way too many for that. But just be aware that this is what this one looks like, and there are similar looking species out there, and they will be out there during the time that we are doing our surveys, as they are active from as early as May through October. And we saw some especially early this year with the warm temperatures. Appearing very similar to our American bumblebee, we also have in our area the southern plains bumblebee, Bombus fraternus, flying from March through November-ish, and she has a little bit larger of a dark segment on the back of the abdomen as compared to the American bumblebee, but she's also a possibility to see out on our surveys. The western bumblebee has several different color patterns, but most noticeably, many of those patterns include a white rear of the abdomen, so a little white tail, it looks like. And these are pretty exciting to see because they have been spotted in urban areas like Fort Collins before, and these are a tier 1 species. There are several species of skipper butterflies on this list as well. Many butterflies are tightly associated in their location with their host plant. So this skipper in particular, Otto, is associated with little blue stem. So if you're out in the field and you're worried that you may have a sensitive species of skipper, if you are not seeing that little blue stem host plant, it is less likely that you are seeing one of these species. Here's what one looks like, just to be aware. The western regal fritillary is a subspecies of fritillary that we have in our area that is a little bit different from the other fritillaries that you will see. Those hind wings are quite a bit darker, so you may notice the difference there. They are somewhat large as well. These guys feed on viola species as caterpillars, so that's things like violets and related flowers. So again, tightly associated in range with those host plants. Generally, we see those from early June to early August. The white-shouldered bumblebee gets its name for how it looks, which is helpful. That first segment on the thorax is quite pale compared to the bright yellow of the abdomen that we normally see. We typically see these guys from April to October, and they can look a little bit similar to some of the other bumblebees that we have in the area, but that first thorax segment being very pale in coloration is a nice giveaway for these guys. The silvery blue butterfly is also in our area. It is a tier 2 species of greatest conservation need, and I would personally have great amount of difficulty identifying this as opposed to some of the other blues that we see in the field that look very similar in my opinion. So these iridescent silvery blue wings on a very small butterfly, we see these quite commonly, this family quite commonly out on our surveys. So always be very delicate, of course, with these tiny butterflies. This one in particular, we want to look out for on the pea family as its host. The tawny-edged skipper is in our area as well as a tier 2 species. It has somewhat unique markings on the wings that can help identify it. It also feeds on a host of panic grass, is the common name. So we find this in grassy areas mainly, from June through August. The golden northern bumblebee, Bombus fervidus, is going to be the most difficult to tell from both the American bumblebee and the white-shouldered bumblebee, in cases where maybe it's a little bit more pale than some of the others. And it is a tier 2 species found in this area from June through August. And it also has that short little black final segment on the abdomen. The long dash skipper is also in our area as a tier 2 species. It mostly feeds on poa grasses. Some of the common names for that include bluegrass or meadowgrass or similar grasses are important for their life cycle for their caterpillars. So tightly tied to host plant distribution as well, and have sort of a curved band with the equal sized yellow spots on their wings that may be unique on the hind wing compared to some of the other skippers that we've looked at on this list. Bombus insularis is another threatened species in our region, and it is a cuckoo bee, the indiscriminate cuckoo bee, because it does nest parasitize several different other species of bumblebees, where the queen will go in, she'll lay her eggs in another bumblebee queen's nest and let her raise them. So because of that, she doesn't have to provision her young with any pollen or resources, so she doesn't have those pollen baskets the way that most other bumblebees do. Um, we do have other cuckoo bumblebees in this area, and you'll notice that she'll be a little bit different because her abdomen will be mostly black. This would be very exciting to see. We usually see sightings of this from March to October. She is listed as a species of greatest information need. Because we are seeing a decline of bumblebees as a general trend, uh, bumblebees that need other bumblebees as their hosts are typically especially threatened. But we just don't have the data to back up where they're at and what their status is. So this is a greatest information needs species in our area. The last bee on our list of species I'd like you to be aware of in our area is the interrupted Xeromolecta bee as one of the common names. It can also be heard to be referred as the interrupted digger cuckoo bee, because it is a cuckoo bee in that it will lay its eggs in other bees' nests, and usually other members of the digger bee group. So both of those common names are used. It has been recently reclassified, so the new genus is Brachymolecta. You may also see it as Xeromolecta. So both of these names may be present if you're looking up more information about this bee. I would absolutely love to see this bee. I have never seen one, and I would love to. They are bright orange with this geometric pattern on their back, dark smoky wings. They will also not have pollen baskets. She doesn't need them. And may be somewhat similar to wasps in size and shape. So do be on the lookout to not confuse it with a wasp. Thank you so much for sticking through that list of species of conservation need in our area. At the end of your survey, hopefully you've seen something cool and enjoyed yourself. Please also remember to remove any seeds that have been caught on your butterfly nets from the plants. We don't want to spread any invasive species. Please also check things like your shoes, especially shoelaces, I find, can pick up a lot of seeds as we're running through the grass. And make sure that we're not spreading any plants that are not welcome in that area. Especially in developed areas where disturbance is pretty common, we do find a lot of invasive plants. So just be mindful of that. Remember to upload your photos to iNaturalist. You don't have to take your photos for the survey through the iNaturalist app. There is always an option to upload from your photos. Upload a picture of your data sheet as well, either to our iNaturalist project where I can see it, or email it to me directly. Either is fine. Remember to sanitize your equipment, especially if you are moving between different sites. If you're like, I'm really excited about this project and I want to go to Boulder and Longmont, fantastic. Just remember to sanitize equipment in between. Um, Many of us have heard about the varroa mites that are affecting honeybee communities recently. So we do want to make sure that we are preventing the spread of any parasites between insects as well in those vials that we're collecting them in for photographs. We recommend usually isopropyl alcohol, so just the same as what you'd have in your bathroom cabinet at home. Or if you have unscented dish soap at home to wash with just soap and water, make sure that everything dries completely before collecting another insect in that vial. We don't need to make them wet and prolong that recovery period from being cold to let them fly away as soon as they're ready. Your next step as a volunteer is, of course, to watch the remainder of the videos in this series, breaking down the different groups of pollinators. But what do you do once you've watched all the training videos that we've provided? Now what? I want to make sure that we have completed online training through Native Bee Watch. They have a tried and tested method for bee identification as far as putting all the many, many types of bees that we have here into morpho groups or groups based on how they look. So this is something that they have published research on before. So we want to make sure that we're using their methodology to make sure, one, our data is comparable, and two, that it's robust. So we are using the CSU Native Bee Watch program for bee identification. So watch their video. Their training is about 2 hours. It's a video online. So you can, if you're the type of person who wants to watch that at 1.5 speed, you're welcome to do that. And they also ask that you complete a quiz. We are actually a huge fan of this quiz, and we would love for you to also follow their protocol of scoring at least an 85% on that quiz. And it's mostly going to look at things like flies that look like bees versus the types of bees that you've learned to identify. So things like that are going to be really important when you're out taking your survey. So please go ahead and complete that quiz with an 85%, just like you would if you were volunteering with Native Bee Watch. Make sure you've made that account on iNaturalist if you don't already have an account to submit those photos. Make sure you join our project at Neighborhood Nature Lab. And if you need access to additional blank data sheets, we have the PDF listed below in the video description. And if you need some more, please do email me if you have any difficulties with printing or anything like that. Please reach out to me for any reason, really. If you're having any difficulty, have questions about any of this, or if you're not sure if this survey is the one for you, if it's not the one you want to volunteer with, if you want to find something similar, I'm happy to connect you with other resources as well. Finally, I'm going to leave you with a reminder to identify to the level that you are confident during your surveys. So if that means writing down something as an unknown beetle because you're not sure, maybe it was a bee or a wasp, I'm not sure, that's perfectly all right. Guessing leaves us with some more unreliable data, and we want to show off what community science can do. What we're going to cover in this series of videos is going to be a lot of information. We've already covered a lot of information just looking at the species of conservation need in our area. So I realize that this is going to be a lot of information and a lot of time that you've invested into us to complete this training, and a lot to keep track of. So first of all, we thank you, we appreciate you, and a part of this first year of surveys is also figuring out what works best. There's tons of surveys specifically out there for things like bumblebees, and we're trying to bite off a big chunk here when we're talking about tackling all these different taxa of pollinators, all 6 of these major groups. So we're still figuring out what works best. We welcome your feedback, and we thank you so much for continuing your training for the Neighborhood Nature Lab.